So today I'm going to talk to us about meiosis. So a little review on chromosomes to start with. So as we all know, genes are located on chromosomes. So for us, for diploid organisms like us, we have two copies of each chromosome. So two copies of each chromosome in every cell, and thus two copies of all the genes on each chromosome in every cell. Homologous chromosomes are the two copies. So you inherited one version from mama and one version from papa. So that's why you have two. Fusion of the sperm and egg brings one version of each chromosome, and so you end up with two of each. So each chromosome, again, is a long single DNA molecule. Chromosomes have many genes along the length of the chromosome. So we have about 20,000 genes in the human genome. We have 23 chromosomes. So if they're equally distributed on the chromosomes, there'd be a little less than 1,000 genes on each chromosome. However, some are bigger and some are smaller. But there's hundreds of genes on every chromosome. And as you know, the chromosomes are packaged on histone proteins wound around them. And so they're packaged down into smaller states so that they can fit into the nucleus. The term gene locus refers to where a particular gene is along the length of a chromosome. So the location or locus, the gene locus for any gene, is the same for the version of that gene on the paternal homolog and the maternal homolog. So if we have a gene A and on the maternal homolog on the mama chromosome, it's 1,500,000 bases from the end. So the version of gene A on the paternal or papa homolog is also 1 million bases from the end as well. So the location of genes is the same on the two homologs. And because of that, the order of genes is the same. So if this is the order of genes on a particular chromosome, gene A, B, C, and D, it's the same exact order and orientation on the paternal. So two homologs have the same order of genes along their length. Now, the version of the gene that's on mama versus papa chromosome might be different. And so different versions of a gene are called an allele. And the allele inherited from mama might be different than the allele inherited from papa for a particular gene. So for instance, here gene A, the maternal version has a capital A, so they have the dominant version of that gene, whereas on the paternal version, the papa chromosome, there's a little a, so it has the recessive version of that gene. So the two alleles that you get can be different. You can get one from mama and a different one from papa, or they could be the same. You could get two dominant alleles, just depends. What's the difference between two alleles? So here I'm talking about a dominant and recessive allele, but what's actually different with the DNA of those two genes? Think about that for a second. So the difference is the sequence. So we talked about how the sequence of a gene determines the sequence of the amino acids that make the protein. So different alleles differ by some sequence. It can be as little as a single nucleotide difference. And then what that does to the protein and the protein function will determine whether that has a dominant or recessive type of function. And there are other different versions of changes in function of genes. So what is the diploid number in humans? Do you know that? You probably do. So diploid number in humans is 46. Then what's the haploid number? Correct. The haploid number is 23. So in almost all of your cells, you have 46 chromosomes. 23 of those chromosomes you inherited from mama, and 23 of those chromosomes you inherited from papa. A karyotype, which is shown here in this figure, is a display of the chromosomes from an individual. So they take a picture of the chromosomes in mitosis. They do it in mitosis because they're the most condensed, so they're more easy to see. So the bow tie versions are easier to take a picture of. And then they align chromosomes 1, the 2 versions here, chromosome 2, 2 versions, chromosome 3. So it's a display of the different chromosomes from an individual, and they can be stained, and things can be recognized due to the staining. So a karyotype is just an array of the chromosomes from an individual. So who can tell me what the sex of this individual is? Pause the recording and figure it out. Everyone got it? And you are correct. So this individual has both an X and a Y chromosome, which in human sex determination means you're male. So X and Y is male. Females would have 2 X's and no Y. All right, so this chapter we're talking about meiosis, but let's just think about the differences between mitosis and meiosis. So overall, mitosis is the division of cells making 2 exactly the same daughter cells. So mitosis starts from the fertilized egg and then makes all the cells of the body. So that's how new cells are made, is by mitosis, and they are exact copies of each other. Meiosis, which we're talking about here in this chapter, is a specific type of division that yields haploid cells. And the types of haploid cells that it yields are eggs and sperm. So for sexual reproduction during fertilization, you want an egg cell from mama and a sperm cell from papa to come together and form a normal diploid cell. So you need 2 haploid cells to fuse to make a diploid. So the sperm cells are haploid and the egg cells are haploid. Those haploid cells are made by the process of meiotic division. So meiosis makes haploid gamete cells, so egg cells and sperm cells. That idea of sexual reproduction, so sperm and egg fusing, which is really important for genetic diversity, that requires this meiotic division so that you can make haploid cells and they can come back together. And we'll talk in another section of the chapter about how important sexual reproduction is to maintain diversity in the population. All right, so let's review kind of the overall meiosis goal. So meiosis starts with a diploid cell. So here's our normal diploid cell. After we go through all the steps in meiosis, we yield haploid gametes. So in this cell, we have we have 2 chromosomes, one version, the maternal version and the paternal version. We go through meiosis and we end up with cells with only one version. So either the maternal version or the paternal version. So we end up with cells with the haploid number of chromosomes. It does that through 2 divisions. So first, the chromosomes are duplicated. That happens before division. So here we have our duplicated chromosomes. We divide once, separating the 2 homologs. We divide again, separating the 2 chromatid. And so we end up with 4 cells that only have half the number of chromosomes as what we started with. Meiosis 1 is the division, the first division that separates the 2 homologs. And so now these cells are haploid. It's only got 1 chromosome. This has only got 1 chromosome, though that chromosome is still duplicated, so it still has 2 DNA molecules attached at the centromere. And then meiosis 2 separates those 2 sister chromatid, yielding the final output, which is 4 cells, each cell with only 1 chromosome, in this case, which is the haploid number of chromosomes. So meiosis overall, there's only 1 round of DNA replication, but 2 rounds of division, so you end up with half the number of DNA molecules that you started with. A little terminology for meiosis that we've already been using. So first is homologous chromosomes, and so you have 2 versions of each. One you got from papa, and one you got from mama. So the maternal and the paternal versions of the chromosomes. Sister chromatids that we talked about in mitosis, again, are the 2 exact copies of the chromosome. So during DNA replication, we make an exact copy. We leave those copies attached at the centromere, and so those 2 exact copies are called sister chromatid. And then in meiosis, we talk sometimes about non-sister chromatid, and that's 1 DNA molecule from one homolog as compared to a DNA molecule from the other homolog. And so the non-sister chromatid are not exact copies of each other. So sister chromatid are exact copies, same exact sequence along the whole length. The non-sister chromatid are not exact copies. They have the same order of genes, A, B, C, D, etc., but they could be different because you could have a big A from mama and a little a from papa. So it's the same order of genes, but not necessarily the same collection of alleles on the two, so they can be different. In this cell, just thinking about haploid and diploid, so the diploid number is always just the total number of chromosomes. So you can do that just by counting them up. 1, 2, 3, 4, 5, 6. So we have 6 chromosomes total. So the diploid number is 6. The haploid number would be half that. And so it would be 3. And that's because we have, 2 big chromosomes, 2 medium-sized chromosomes, and 2 little. So if we had one version of each, we'd have 1 big, 1 medium, and 1 little. So that's the haploid number of 3. So let's talk about what goes on in meiosis. And things are quite different at the beginning of meiosis than what we talked about in mitosis. So the first thing that happens in prophase 1, so in meiosis we have 2 division. All the steps in the first division are called prophase 1, metaphase 1, anaphase 1. And in the second division it would be called prophase 2, metaphase 2, anaphase 2. So most of the differences that we're going to talk about between meiosis and mitosis are in the first division. So in prophase 1, one thing that happens that doesn't happen in mitosis is synapsis, where the 2 homologous chromosomes line up and pair to each other. So the 2 chromosomes kind of stick together and they align gene to gene. So that doesn't happen in mitosis. So here in meiosis we have the 2 big chromosomes lining up next to each other. We have the 2 medium-sized chromosomes lining up and the 2 small ones. That collection of the 2 homologs lined up along each other is called a tetrad. The next thing that can happen in prophase 1, and that happens on average for every chromosome in a meiotic division, is exchange of segments between the maternal and the paternal version. So that's shown here. Those sites of crossing over are called chiasmata, and you can see that here we've switched the blue and red ends of this chromosome, and you can see that in the output down here. So here's a chromosome that has a blue end and a red end. So we were actually exchanging material. We cut and paste different parts of the chromosomes and put it on the homolog. And so crossing over again is exchange of segments between the maternal and the paternal version. And so that yields the red end here, which means you have some of mama genes on the chromosome that you originally inherited from papa, and vice versa. The next thing that happens, which is also different than what goes on in mitosis, is in metaphase 1. Now the homologous pairs line up along the metaphase plate. So instead of each chromosome lining up by itself, now the 2 pairs align next to each other. So they've lined up, they've crossed over in prophase, and now they all line up on the metaphase plate next to each other. And spindle microtubules, in this case, attach to the different homologs. So this pole is attaching to the red homolog, whereas this pole is attaching to the blue. So these 2 are stuck together, and the red one's being pulled this way, and the blue one's being pulled this way. And so that's going to set up a situation where you can separate the 2 homologs in the next step, which is anaphase. So the attachment that we had in metaphase is released, and now one homolog, in this case the mama version, is going in this direction, papa version is going in this direction. And so in anaphase 1, we separate the 2 homologs. So the maternal version will go one way, paternal in the other way. And for each chromosome, it's random whether it's maternal or paternal go to either end. So the homologs migrate to opposite spindle poles. And then in the last step, telophase of meiosis 1, so telophase 1, the nuclear envelope reforms, and we end up at the end of meiosis 1 with 2 cells, but now those cells only have half the number of chromosomes. So we only have 3 chromosomes in this cell, 3 chromosomes in this cell, we started with 6, so now we're down to haploid cells. But notice that the chromosomes in these haploid cells still have the duplicated DNA molecules. So each chromosome now still has 2 DNA molecules. Cytokinesis, again, is the splitting off of the 2 cells. Then the second division is meiosis II. And the main thing that happens in meiosis II is we now separate the 2 homologs. I mean, not the homologs. We now separate the 2 sister chromatids. So each chromosome has 2 DNA molecules, and we're going to separate those 2 DNA molecules into 2 different daughter cells. So that's the overall goal of meiosis II, is to take those chromosomes and separate the 2 DNA molecules to opposite cells. This process proceeds, this looks very much like mitosis, but you're beginning with haploid cells. You only have 3 chromosomes instead of the 6 that you would normally have in this cell. So meiosis II is like mitosis, except that you're starting with only half the number of chromosomes, but you are just separating the sister chromatid as you would in mitosis. And at the end of telophase and cytokinesis 2, you end up with 4 cells, because you had 2 cells at the end of meiosis 1. Both of those divided again. So now we have 1, 2, 3, 4 cells, and each of those cells only has 1 version of each chromosome. So all of those cells are haploid. Because of crossing over, the chromosomes in each of the 4 cells is different, because some have red bodies and blue tips, and vice versa. Some would have just all blue or all red. So you end up with 4 different haploid cells as the product of meiosis. And a good explanation of that, and a better way to visualize that, is to look at the meiosis video. So I recommend you look at the meiosis video, and that really will make it clear why you end up with 4 different haploid cells. Okay, I want to finish up here by talking about or reviewing the differences between mitosis and meiosis. And so there's kind of 3 main events. Synapsis and crossing over. So in the beginning of meiosis, the 2 homologs align next to each other and exchange segments of DNA between the 2 homologs. So that occurs in meiosis. That does not occur in mitosis. During metaphase, metaphase 1 specifically of meiosis, the 2 homologs line up next to each other, whereas in metaphase of mitosis, they don't. They line up by themselves. And so, again, mitosis, they line up by themselves. And then the 2 homologs are separated in anaphase. And so meiosis yields 2 cells at the end of meiosis 1 that only have 3 chromosomes instead of the 6 that it started with. And so note that all the differences between mitosis and meiosis all occur during meiosis 1. Meiosis 2 is very similar to mitosis, but it's starting off with only half the number of chromosomes. A couple figures just to highlight those differences between mitosis and meiosis. So again, in meiosis, we line up the 2 chromosomes next to each other through synapsis, so they zipper up gene to gene, and then some of the DNA material is exchanged between the 2 homologs. The next thing is that the 2 homologs line up next to each other along the metaphase plate, and as in mitosis, they all line up by themselves. So here they're all lined up by themselves. In mitosis, you're just separating the sister chromatid, but in meiosis, you are aligning the 2 homologs next to each other, so you can separate them to 2 daughter cells. And then, then at the end of meiosis 1, you've separated the 2 homologs so that you end up with daughter cells that only have 3 chromosomes instead of 6. So here in mitosis, the end is 6 chromosomes in this cartoon, so that's the diploid number, whereas these only have 3 chromosomes, and 3 would be the haploid number for this cartoon cell. And I think that's it for this video. There's going to be some questions looking at the difference between mitosis and meiosis as an assignment in Moodle. Thanks.